Hello, everyone. Welcome to Chapter 12 podcast. We're going to cover two podcasts with this material. And the first podcast, we're going to talk about chromosomes and their structures and how you compare different chromosomes. And in this second podcast, it'll cover Objective 3 from the note taking guide. And that's going to talk about cell division and more precisely mitosis. But first, let's think about just the general term of cell division. Cell division, as the name implies, you start off with a single cell, and then this single cell divides into two cells. Okay? Usually. Now, when this can occur by two different cell division mechanisms. This first is mitosis. I'm just going to write MIT for mitosis. And then our second one would be meiosis. And I'm going to draw two lines here. And at the end of meiosis, we end up with 4 cells. And so let's go ahead and write meiosis here. And the reason I drew 2 lines is we'll learn in a few chapters from now that meiosis involves 2 separate divisions. Some of the differences are that mitosis we typically think of as being found in somatic cells. And meiosis we typically associate with being in gametic cells or in our gametes. Somatic cells are the cells that are found in every part of our body or any organism's body. These are the cells that when they divide, they create genetic clones. So for instance, when your skin cells divide, they start off having 2 copies of each chromosome. And when they end up with, at the end of mitosis cell division, 2 cells also with 2 copies of each of the chromosomes. And they're identical. There's very little variation. Every now and then you get a mutation, you get some variation. But mitosis is set up in such a way that you should get 2 identical cells. Meiosis, we'll learn that it has a very similar setup of cell division, but it goes through 2 separate linked cell divisions, and you end up with 4 cells, each with just 1 copy of the chromosome in. So you go from 2 copies to 1 copy. And these 4 copies are genetically quite different than what the original parental cell was. Now we're going to come back to meiosis later. So we'll put a check there. And we're going to focus most of our time right here on our good friend mitosis. Now when we think about a cell division, mitosis or meiosis, but again we're focusing on mitosis, what we're really thinking about is how do we copy the chromosomes and then segregate them into 2 separate cells. So let's take a step back and let's consider the chromosome. If we took a cell, any old cell, and we lysed the cell membrane and released all of the chromosomes, you would see something like this. The cells would be all thrown about. The chromosomes would be all thrown about, and it would be really hard to decipher what you're looking at. But what geneticists will do is they'll take this here mess of chromosomes, and then they will organize them. And they will organize them in something like this. Okay, now in this karyotype, the collection of the chromosomes, how are they organized? They're organized by size and the number of bands, and the location of the bands, and the position of the bands. Okay, so we see right along here, number 1's hard to look at because it's kind of curved here, but let's look at 2. You can see that this chromosome here, and this chromosome is the same length. In addition, if you look at these dark bands here, they're the same location on both chromosomes, and they're the same thickness. And so chromosome 1 and 2 look about the same size, but the banding is different. And this banding pattern we see here is the same in every human. So this doesn't change from human to human. And we call this a karyotype. And let's go ahead and write the word karyotype here, just so you know what I'm talking about when we mention it later on, karyotype. So, just to familiarize yourself with what we're looking at, each one that you see here is a separate chromosome. So this is a separate chromosome. This is a separate chromosome. This one, this one, this one, this one, all the way down here. And if you count them, you would see that in humans, anyhow, this is not true for all organisms, but we're looking at a human karyotype, that in humans we have 46 chromosomes. You could also look at this as us having, I'm going to have to try to squeeze this in here, 23 pairs of chromosomes. Because we can see we have 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, and all the way down the line. A couple other things we should define here. In humans, we have 2 sex chromosomes. Either 2 X chromosomes, as we see here, or you'll have just one of these X chromosomes and a Y chromosome. And the Y chromosome is a really puny little old chromosome. It looks something like that in comparison to this. But I'm going to go and cross this out because I don't want this to get confused with that yet. So we have these sex chromosomes. And sometimes when I abbreviate chromosomes, I'll just write an X and then a SOME for chromosome. So if you ever see me write an X and then a chromosome and then some, I'm talking about chromosomes. All of these others, I'm not going to circle it because it'll get messy, but all of these others, 1 through 22, we call these autosomes. And I just got thinking, when I abbreviated my chromosome here, I did it right by the X chromosome, and I don't want that to confuse you later if you're looking at this. So let's just go ahead and write out chromosome here. Okay, now let's look at the chromosomes in a little bit more detail. There's a few things I'd like to point out to you. The first is that each of these chromosomes are simply a single large, long piece of DNA, a single DNA molecule. So for instance, right here, if you were able to look at this under fine microscope, you would be able to see that this is just one large DNA molecule. And so is this. And as we've said in our previous podcast, this large piece of DNA, the chromosome here, is broken up into genes. Each of these chromosomes may have thousands of genes on them. The larger ones up here are going to have more, of course, and then these tiny ones down here will have fewer. Now let's ask the next question. And this next question is, where do these chromosomes come from? We get these chromosomes from our parents. And this isn't just in humans, of course. This is all organisms do it in a very similar way. Now, when we talk about fungi versus plants versus humans, there are some differences. But right now, we're just going to sort of focus on this model here. So each of these chromosomes, let's pick on chromosome 14 here. We know we get one of our chromosomes, say it's this one, from our mom. And then we get the other one from our dad. And it could be either one. We don't really know which one comes from whom by looking at this karyotype. And this is true for each of the chromosomes. We get one copy from mom, one copy from dad. And we end up, as we see here, with 2 copies of each. Now, let's find another chromosome to pick on. We'll pick on chromosome 6. When we consider these 2 chromosomes together, we call them homologous chromosomes. And in retrospect, I should have picked on 12 so I could have a little bit more space to work on. So, but it's true for all of them. So the 2 1s together, the 2 2s together, the 2 3s together, 2 4s together, the 2 5s together, and all the way down are all examples of what we call homologous chromosomes. prefix homo means same, and logos means the body here, the chromosome, the structure here. So they are the same. And when we say they're the same, what we mean is that if you look at the order of the genes, say from this chromosome all the way down, it's the same genes on the other one all the way down. So structurally they're the same. They're the same length. They contain the same number of nucleotides. They contain the same number of genes and the same order of those genes. The only difference is we get one from mom and one from dad. There could be mutations in one or the other, and that's what helps generate some of the diversity that we see within any population. But we say they're homologous because it's the same genes. Likewise, if we looked at one of these from chromosome 4 and one of these from chromosome 5, we would call those non-homologous chromosomes. The next thing I want to talk about when we look at this karyotype, and the next one I'm going to show in just a moment, is how can you tell by looking at these if they are replicated or non-replicated DNA. We haven't talked about the cell cycle yet, but let's just give you a brief preview to that. With the cell cycle, I'm just going to be very simplistic about this until we get to the next podcast. We have one part of the cell cycle called interphase. And for now, I'm just going to put an A here. And during interphase, several things happen. But one of the big things that happens is that the DNA is copied. So for a chromosome will go from a single chromosome to a chromosome like so, that's attached in the middle by what we call the centromere. And I say attached in the middle, that's not always true. It's not always attached in the middle. Sometimes it can be here at the top or at the bottom. But it's true, wherever it's attached, it's always the same for that chromosome. So this would be unreplicated, and this would be replicated. This middle part, just in case you're wondering why I didn't complete the circle, this is the part where mitosis occurs. And again, we'll spend much more time on that in the next podcast. But just recognize that during cell division, during the large phase of interphase, the DNA is copied. We go from a single chromosome to 2 chromosomes attached. So over here, this karyotype represents a single chromosome, right? Because we have one here, we have one here. All of these that I'm tracing with the pen here are single chromosomes. To help you understand that better, let's look at a karyotype where we have replicated chromosomes shown here. So we see here we have chromosome 1, 2, 3, just set up the exact same way we see over here. But the difference is, let's see which one we want to draw on. So we still have our one single piece of DNA, the first chromosome, coming down here. And then this is the copied one over here, coming down here. And they are attached here at the centromere. So I want to go over a couple things when we're looking at this. This area of constriction is the centromere. We don't see it over here, but it is there, or I should say it's more difficult to see, but they are here. So on this side, here's our centromere. Much harder to see, but every chromosome has them. It's just easy to see after they've been replicated because you can see where the 2 chromosomes are attached. All right, one other thing that we want to cover here, and this is always really confusing, and that is how do we count the chromosomes? And again, we'll cover this a little bit more when we talk about mitosis, but I think it's good to introduce it here. Here, if we count these number of chromosomes, we would say we have 46, as we said. Over here, you might be tempted to say we have 92 chromosomes. But as long as the chromosomes are attached in this area here at the centromere, and I should spell out centromere so you know what I'm talking about from your notes. As long as they're attached, we call them a single chromosome. So as long as they are in this attached state here, we would still say that this karyotype contains 46 chromosomes. But since we're scientists, we have to have another name to refer to each of these. And this is where I think it gets kind of confusing. And so I think I always got confused when I thought about these three terms. Chromosome, chromatid, I'm sorry, let's say chromatin here. And the other term that I would often get confused would be chromatid. And this is a really good thing to make some flashcards with till we get used to these terms. Chromosomes and chromatin, the first two here, they're nearly identical. They just have a different part of the cell cycle they're associated with. Up here, remember we said we had interphase here and mitosis here. During interphase, this chromosome is called chromatin. So over here, I'm just going to write I, so that you know that during interphase we call it chromatin. Now why do we do that? Well, it's not strictly to confuse people, but it's because we're talking about the state of the DNA with the protein packaged around it. During interphase, this DNA is much less condensed. It's much harder to see under a microscope. You would need a really high-powered electron microscope to see it. And we can talk later about why that's the case. But during mitosis, but we're talking about mitosis here, this chromatin becomes highly condensed. So here I'm just going to write M. And that's what we see in these two karyotypes. These are chromosomes. This is what we mean by a highly condensed structure that's tightly packaged together. So both chromosomes and chromatin, by definition, are the DNA and the proteins that are associated with that DNA. And the only difference is where you would find them during the cell cycle. Chromatid, we only talk about this term, well, I wouldn't say only. We refer to each of these as chromatids. So here we have a chromatid, and attached to it after replication is another chromatid. Same here and same here, and all the way across here. We even will call these as a generic term, even though they are chromosomes at this stage, we might call them chromatids too. That's something that's often exchanged. So as we're looking at this, now I have another question for you, and that is, as you look at this karyotype, how many chromatids do you see? And on this karyotype, how many chromatids do you see? And over here, we have, on the left side, we have 46 chromatids. And over here, remember we said we had 46 chromosomes, 23 pairs. That's because the chromosomes are attached. But each of these chromosomes contain 2 chromatids. So over here, we would say we have 92 chromatids. So I'm going to go to a new page here, and I just want to review some of these terms and how we count them. Okay, so let's do some drawings. So let's draw a chromosome here. And once it's been replicated, let's draw it again. And because, remember we talked about in the last chapter, that replication is semi-conservative, and what that happens, just as review, is we take this chromosome and this, and they're each used as a template. So the new one, I'm going to draw in as red, just to signify that this is the new copied one. And I mean for these to be the exact same size, just that by freehanding it might not be perfect. And so we have to do with the other one as well, because remember, both of these are going to be replicated. Remember, in any given cell, let's just say this came from our, skin cells. One of these originally came from one parent and the other came from our other parent. Remember, they're the exact same number of genes in the exact same order on both sides. In this very simple drawing here, we can say, in fact, why don't we do this? Let's write the word chromosome and chromatid. How many chromosomes do we have here? The answer is 2. How many chromosomes do we have here? The answer is still 2, because they're attached by this centromere. Over here, we'll say how many chromatids do we have? We'll say we have 2. And over here, how many do we have? We would say we have 4. Each one of these is a separate chromatid. And if you come back to the next podcast, we'll explain how we go from here to the next stage, where these chromatids will come apart. Now, often what we'll call these two here, I should have said this on the last slide, but while it's on my mind, I want to make sure I mention it, is we'll call them, these two together, we call them sister chromatids. And sister chromatids just means that they are identical to each other. Sister chromatids. And they're identical because they were just copied. All right, that ends our second objective for this chapter. Don't forget to watch the next podcast. That's going to talk about mitosis. And we'll see you in class. Bye.